Hello everyone, welcome back to my channel. And in this lecture, we are going to discuss about the dark reactions of the photosynthetic process, which are also known as the Kelvin cycle, or they're also known as the C3 cycle. Why they are called by these names, that is also what we are going to look at. So photosynthesis has been primarily divided into two types of reactions. The first one are the light reactions and the second one are the dark reactions. Light reactions we've already discussed, where we saw that these kind of reactions are dependent on light and these happen in the thylakoid membrane. At the end of the light reaction, there are certain high energy molecules such as ATP and NADPH, which are being produced. And these are the ones that are then used by the dark cycle in the stroma. So let us try to understand all this now. This video is primarily about the dark reactions, and these dark reactions, they are light independent, which means that they can occur in light or in the dark. So light presence is not mandatory for these kind of reactions. Now, these dark reactions, they occur in the stroma of the chloroplast. And these reactions use the chemical energy that is being produced in the light reactions, that is the ATP and the NADPH molecules that we just discussed. And these are used to reduce the carbon dioxide to the carbohydrate molecules. So all these processes, they are going hand in hand. So the light reactions are basically producing energy, which can be utilized in the dark reactions to produce carbohydrate molecules, the sugar molecules, which we are consuming right now. And in this process, the carbon dioxide is converted to the sugar molecule by entering into the Calvin cycle. Now, this Calvin cycle is called so because it's named after its inventor, which is Melvin Calvin. Also, this is known as C3 cycle because the first stable compound of this pathway is a 3-carbon compound, which we will look in the next slide. But before we actually dive into understanding this cycle, let's just try to revise everything that is happening inside the plant cell, specifically the chloroplast. So you see, these are the thylakoids. You can see the grana basically present inside the chloroplast. So when light falls onto it, there are certain series of electron transport chain happening, leading to the production of ATP and NADPH molecule. And also in this process, a water molecule is being split to form oxygen, which we are breathing right now. These ATP and the NADPH molecules then enter into the Calvin cycle, which is the dark cycle. We are concerned about that in this lecture. And that leads to the production of sugar molecules finally. And also after the end of this reaction, there are the products, broken down products formed, which are again utilized by the light reactions to convert them back into the ATP and NADPH, which can again be used in the Calvin cycle. So let us try to see that what exactly this Calvin cycle is doing and how it is actually taking place. So in the Calvin cycle, carbon dioxide is entering. So the carbon dioxide, we all say that, okay, the plants, they use carbon dioxide for production of food. So this is where the carbon dioxide is being utilized. Now, there are a total of 3 stages of the dark cycle. The first stage is known as the carbon fixation. Second stage is known as reduction. And the third stage is known as regeneration. So CRR. There is carbon fixation, reduction, and regeneration. So this whole cycle starts with a 5-carbon molecule known as RuBP, which stands for ribulose 1,5-bis phosphate. So we are starting with 3 molecules of this 5-carbon compound. So in total, we are starting with 15 carbons. Just keep that in mind. So this ribulose 1,5-bis phosphate is a molecule which is having 2 carbon molecules, 2 phosphate molecules. One is attached to the carbon number 1 and the second one is attached to carbon number 5. There is something that we are going to do with these phosphate molecules. So keep on understanding as I explain the process. So we are starting with 3 molecules of this RuBP molecule, and this RuBP is having 5 carbons. So a total of 15 carbons for now. Now, there is this enzyme called Rubisco, which stands for ribulose bisphosphate carboxylase oxygenase. This enzyme is having dual activity, which means that it can act as a carboxylase enzyme as well, and it can also act as an oxygenase enzyme. Now, what does these two types of enzyme means? So carboxylase is the enzyme that has the affinity for carbon dioxide molecule. On the other hand side, oxygenase is the enzyme that has the affinity for oxygen molecule. So under what condition this Rubisco enzyme is going to show the carboxylase activity and under what condition it's going to show the oxygenase activity, it depends on the concentration of the molecule present around its periphery. So if there is an excess amount of carbon dioxide present in the periphery of this enzyme, then it will show its carboxylase activity. Whereas if there is excess amount of oxygen as compared to carbon dioxide, then this specific enzyme will bind to the oxygen, which by exhibiting its oxygenase activity, and that specific process is known as photorespiration. So that is an altogether different kind of pathway where some additional steps are included, leading to wastage of energy. But here we are concerned about the fixation of the carbon dioxide. Only if the carbon dioxide is going to fix with the Rubisco, then only it can proceed into the Calvin cycle to form the sugar molecules. So in order for this Rubisco to show its carboxylase activity, there needs to be a high concentration of carbon dioxide present in the cell. So let's suppose that there is enough carbon dioxide present. So this Rubisco is showing its carboxylase activity and it's going to bind to the carbon dioxide molecules. Now, because we are starting with 3 molecules, we are going to include 3 carbon dioxide molecules here. So when 3 carbon dioxide molecules are used by the Rubisco enzyme to put onto the RuBP molecules, that is where the carbon fixation comes into play. So this enzyme is going to fix these carbon molecules from the carbon dioxide onto the RuBP. And how it is going to do? So what it's going to do is, it's going to attach to carbon dioxides and going to split the bigger molecules into shorter 3-phosphoglycerate molecules. So you see, we started with 15 carbons and we got 3 carbons from the carbon dioxide. So a total of 18 carbons. And now after the breakdown and all that, there are going to be 6 molecules of 3 carbon molecules, 3 phosphoglycerate. So a total of 18 carbons. So there is some kind of rearrangement going on here. So this 3-phosphoglycerate is the first stable compound of the Calvin cycle. That is why this cycle is also known as C3 cycle, because 3-PGA is the first stable compound of this cycle. Now, the next step is reduction. So there is going to be the reduction of this molecule PGA into glyceraldehyde 3-phosphate. How it is going to do? So there is going to be certain reaction going on where these groups are going to face some changes, and that is going to happen because of the high energy molecules which were provided by the light reactions into the stroma. And those high energy molecules are the ATP molecules, and we have NADPH molecule. Now, this is a strong reducing agent, and that is why it is being used here. Why we have added 6 here? Because we are dealing with 6 molecules of PGA. Each ATP and NADPH is going to act on one 3-PGA molecule. So at the end, we are left with 6 molecules of glyceraldehyde 3-phosphates. Now, we have 6 molecules. Out of this, one glyceraldehyde 3-phosphate molecule is separated from the cycle and it goes towards the formation of glucose molecule. And the remaining 5 molecules, they undergo phosphorylation by the use of ATPs. So the phosphate group from the ATP molecule is going to come and attach to the carbons leading to the formation of RuBP again. So there is regeneration going on here. How it is happening? You see, these are six molecules of 3 carbon molecules, which means that it is a 18 carbon. And then these are going to be rearranged in a way where 3 carbons are exiting this cycle for the formation of glucose, and the remaining 15 carbons, they are phosphorylated and rearranged in the form of RuBP molecules. So again, we have 15 carbons here, you can see. So there is regeneration again. And Rubisco is the most abundant protein present on the earth. Now, this is the final reaction that we get after we have understood the Calvin cycle. So a total of 6 carbon dioxide molecules, 18 ATPs, 12 NADPH, and 12 water molecules, they combine together to form one glucose molecule. Now, why we are writing one here? Because you can see that when we are dealing with 3 molecules of RuBP, we are starting with that, we are getting half molecule of glucose from one G3P molecule. So in order to make one full glucose molecule, we need to have 2 G3P molecules here. So that basically doubles up everything that we have been doing. So it's going to be 6 molecules here, 6 molecules here, this is going to be 12, and this is going to be 12 again, and then we are getting one glucose molecule. Thank you so much.